Okay, looks like we're about to get started. So with that, I'd like to welcome everybody to the Office of the Chief Scientist Science Seminar Series. For those of you who don't know me, my name is Vanessa Swarbrick, and I'm a Senior Science Advisor with the Office of the Chief Scientist and your host for this winter seminar series. On behalf of our entire team, welcome to today's event. I'd like to start by respectfully acknowledging that in Alberta, we are on the traditional gathering places and home to the peoples of Treaty 4, Treaty 6, Treaty 7, Treaty 8, Treaty 10, and the Métis Nation. I would like to acknowledge the diverse Indigenous peoples whose footsteps have shaped this territory and whose histories, languages, and culture continue to influence our communities today. Wherever you find yourself today, I encourage you to recognize and acknowledge the relationship that First Nations, Inuit, and Métis have with the land upon which we all live, work, and learn. Now, in keeping with the tradition of gathering, these science seminars provide an opportunity for us to get together, to exchange knowledge on interesting science happening across Alberta in an open learning and sharing environment. The hope is that through our dialogue today, we are all able to improve our awareness and understanding and to put this knowledge into practice to support evidence-informed decision-making wherever that occurs. As you may have noticed, when you join the call, we are recording today's seminars. Your microphone and video are disabled. However, you can enter your questions or comments at any time during the chat via the chat. The chat is not going to be recorded. After the presentation has concluded, I'll read those questions out in the order that they were received. And with that, I'd like to introduce today's speaker. Today, we are welcoming Dr. Tim Lysyk, who is presenting on the distribution of two expanding tick species, Dermacentor andersoni and Dermacentor variabilis, in Alberta and Western Canada, determined by active sampling. Dr. Lysyk received his BSc in zoology from the U of A, an MS in entomology from South Dakota State University, and a PhD in entomology from North Carolina State University. He was employed as a research scientist with the Canadian government for 30 years, much of it spent at the Lethbridge Research Centre. He retired in 2014, but still maintains an active collaboration with tick researchers across Western Canada. He is an adjunct professor at the University of Calgary Faculty of Veterinary Medicine and is currently the co-editor-in-chief of the Journal of Medical Entomology. And with that, I'll pass the floor over to you, Tim. Welcome.
Thank you very much. Thank you for the opportunity to speak today. I'll be summarizing several years of work that we've done examining the distribution of a couple of species of ticks in Alberta. Specifically, I'll be looking at Dermacentor andersoni, which is the Rocky Mountain wood tick, and Dermacentor variabilis, the American dog tick. The type of work requires a lot of collaboration, and I've been really fortunate to work with some really wonderful people on this. In particular, Sean Durgasoff from Agriculture and Agri-Food Canada, Neil Chilton from the Department of Biology, University of Saskatchewan, and Dr. Catherine Rochon from the Department of Entomology, University of Manitoba. We've had funding from Agriculture and Agri-Food Canada, Alberta Livestock and Meat Agency, Beef Cattle Research Council, Alberta Agriculture and Forestry, and excellent technical support from our technical staff, Christine Himsel-Rainer, Karen Toohey, and Lisa Babby. Been a lot of students involved with this work, and we're also really grateful to the landowners and managers who've led us on to access their properties. There's a number of species of hard ticks in Alberta, as well as a few species that are considered adventitious as they only show up on migrating hosts. The two species I'm going to be talking about that I've got at the top of the list here, the first one, Dermacentor andersoni, has been known to occur in Alberta for an awful long time, and it's probably the most common tick that people will encounter, at least around here. Dermacentor variabilis was not listed as a tick that occurred in Alberta, not even in the most recent Handbook of Ticks of Canada, written by Lindquist et al. It really wasn't mentioned in any of the historic publications, but it does show up in some of the passive surveillance that goes on in the province. In my personal experience, I usually saw it from dogs that had traveled outside the province. It can be easily confused with Dermacentor andersoni. And lastly, for another member of the genus Dermacentor that occurs that can sometimes be confused with andersoni is the winter tick, Dermacentor albopictus. This is widely distributed on moose and other large mammals in North America, and has been actually extensively studied out of the University of Alberta by Dr. Bill Samuel. To recognize tick, it's always important to be able to recognize and know what you're talking about. To recognize ticks, we usually look at the shape of the mouth parts, really to separate them into the different genera. The genus Dermacentor has a very characteristic mouth part. There's a little rectangular plate at the base of a set of palps. This plate is known as the basicapitulum, or basicapituli, and the most characteristic thing about it for this genus is that it has these straight sides. It's rectangular with straight lateral sides. And the second characteristic is that the palps there's two visible palps that you can see from the top. They're about equal in length and relatively short, about equal to the width of this basicapitulum. To get down into the species level, we have to look at flip the tick upside down and look at the spiracle, which is a breathing apparatus that is located behind the hind set of legs. So this is why we worry about looking at these spiracles, because most of these species of ticks, we have the females on the left and the males on the right. They look relatively similar. They're sort of reddish brown with kind of a little white markings and so on them. But if we look at the spiracles, we see that they can be quite different. And so for the winter tick, it's largely basically sort of an oval type of spiracle with quite large goblets in it. Rocky Mountain wood tick has a dorsal prolongation on the spiracle, lots of large-sized goblets, but there's also surrounded by a couple of rows of small, fine pores. And then variabilis also has a dorsal prolongation, which you can't really see in this picture, but it has very, very fine pores scattered throughout, and numerous pores scattered throughout. So that's really how we tell these three species apart. Why we're interested in them is that they do, members of the genus Dermacentor are capable of transmitting a number of pathogens. My own personal interest has largely over the years has focused on the very first item here, tick paralysis, which isn't caused by a pathogenic agent, but it is caused, or like a disease agent. It's really caused by something in the tick saliva, and it occurs in British Columbia, and it can cause lameness and ultimately death if the ticks aren't taken off. And luckily, we don't really see much of that condition in Alberta. The ones that we have seen in the past are rickettsia, which causes Rocky Mountain spotted fever, tularemia, and Colorado tick fever have all been reported from Alberta. There's also some suggestion in Dermacentor andersoni is a demonstrated vector Q fever, but we're not entirely sure how important a role it plays in the transmission of this particular agent. We don't see a lot of these, I don't think, in Alberta these days. They used to be in the past, and so the public health significance of these ticks is probably not great. What we've mainly been interested in and why we did a lot of work on the distribution was because of Anaplasma marginale, which causes bovine anaplasmosis, and that can be transmitted by Dermacentor andersoni. Bovine anaplasmosis is a blood disease of cattle. It causes anemia and mortality in older cattle. Survivors are persistently infected. Years back, it was originally, when we started this work, it was a reportable disease in Canada, but due to periodic outbreaks, it's been downgraded to a notifiable disease. There used to be import testing requirements for cattle coming in from the United States, but these were considered a non-tariff trade barrier by the US, and this requirement was a significant trade irritant. We had demonstrated that anaplasmosis can be transmitted by our native Dermacentor andersoni. And so as a result, transmission risk is related really to the presence of these ticks. So it's important to be able to understand the distribution of these ticks so that we know where this now notifiable disease could possibly occur in Alberta. I'll give you a little bit of a historical perspective on this, and it's always been quite interesting to me, but there wasn't originally an Alberta Rocky Mountain spotted fever survey, and they actually started in 1938 and ran for about 11 years. A couple of series of publications came out which really defined and has been probably been used mostly for defining the distribution of Rocky Mountain wood tick in Alberta. In the east, it appears about 51.4 degrees north, just around equal with Hannah. It used to be thought that it just pretty much went straight across the province, but there were some detections up in Jasper early on. So it gives it this bit of a distribution where it skirts across the province from east to west with this northern extension along the Rocky Mountains. Really, this was the pattern that was reported in subsequent papers. In subsequent papers, for example, this one by Jack Gregson in 1956, where he published on the Exotidae of Canada. He mentioned that Dermacentor andersoni had 105 degrees west as its eastern limit, and that there was a 50-mile gap between these two species, andersoni and variabilis. Variabilis being more in the east than andersoni in the west. Paul Wilkinson, in his 1967 publication on biogeography of Dermacentor in Canada, reaffirmed this 105 degree separation for both species. He indicated the northern limits of nearly 54 degrees for andersoni in western Alberta, and this was a host record from Rocky Mountain goats. He also indicated a northern limit of about 52 degrees north for variabilis, an approximate 500-meter elevation limit. And that's pretty much where our current knowledge stayed until 2013, when Sean Gergesoff, Neil Chilton, and others published their work on range expansion of Dermacentor variabilis. They did a couple of really, I think, useful things in this paper. The first was basically to present collate early information on the two species tick distributions in Western Canada, and collate and put together this map where you can see with the black parts are the black dots represent the historic distribution of variabilis, and the white are the historic distribution of andersoni. And you can see that they're separated around 105 degrees west. So this is where that piece of folklore came from. Secondly, they went out and actively surveyed for ticks throughout Saskatchewan and a little bit in Alberta as well. And their results can be shown here. The black circles are areas where they discovered Dermacentor variabilis, and the gray circles are areas where they discovered both variabilis and andersoni living in the same habitat. And then there's some white Dermacentor andersoni only points in Alberta. And you can see here that this really provides some very clear evidence that variabilis had been moving west past this 105 degree limit. They've recorded it up to about 100 degrees west. So given that the surveys in Alberta were well over 50 years old, our current state of knowledge was quite old, we decided to re-examine the distribution of Rocky Mountain wood tick, Dermacentor andersoni, using standardized sampling. We sought to determine geographic patterns in its prevalence and abundance, and determine the relation of these to environmental variables. We initially started this work during 2011 and 2012 with a focus on Dermacentor andersoni in Alberta, but after this range expansion of variabilis became obvious, we expanded the scope of our studies to include all of the Western provinces, and to also include trying to detect variabilis where it would occur in areas that it hadn't previously. And that work was done a little bit later on, 2014 to 2019. We used a standard sampling program where we would send people out in the field to drag meter square flannel cloths. This takes advantage of the ticks' questing behavior. What they will do when they're host seeking in the spring is climb to the top of vegetation, wait there for a host to come along, and as they feel vibrations, they become activated, hosts brush up against the grass, and the ticks will latch on and start to feed them. And we simulate that with these cloth drags walking through the environment. We had done a bit of work with Catherine Rochon and Glenn Scholes from the US, looking at sampling methods, and basically decided that we would get optimal results if we used about 10 meter quadrats and roughly about 200 quadrats per site. So 10 meters long, roughly you're sampling about 2 kilometers in an area of rangeland. During the early parts of our study, we sampled just in Alberta, and we sampled 86 sites. There's a distribution of the sites that we sampled. You can see we went up to past 52 degrees north. 86 sites in 2011, 89 sites in 2012. Both years, between both years, 75 sites of those were in common that we had sampled both years. And again, we had quite a large distance that we had people walking over this to conduct the sampling. So we did cover quite a bit of territory for the study. What we did find was that tick abundance and prevalence varied among years. Both were greater in 2012 than in 2011. 2012, about 43% of the sites were positive. 2011, only 24% were positive. We found that the tick density varied and prevalence both decreased with latitude. You can see as you move towards from the 49th parallel to moving up north, both density and prevalence decrease. We found that the maximum latitude that we found ticks at was about 51.58, just about 52 degrees. So a little bit farther north than had been seen in the western parts in the past. We did find density also increased as you move eastwards towards Saskatchewan, and prevalence also increased. But what was kind of interesting was that in some of these montane areas along the western border of Alberta, we also did see some areas that had particularly high tick abundance. Again, west is with a little bit of a gap in the middle of the province. And that didn't show up in terms of prevalence. So what we had was fairly patchy distributions along the front range of the Rockies. This pattern reflected the natural subregions in Alberta, with the dry mixed grass region, which is the lightest green, having the highest abundance and prevalence, and the montane subregion, which is in the dark olive here, having relatively high abundance but a lower prevalence, and again indicating patchy distribution. Abundance and prevalence were lowest in the other natural regions, and this was natural subregions, and this was an artificial group for analytical convenience, and contained places like parkland, dry mixed wood, foothills parkland, and northern rescue. We also looked at the geographic pattern in relation to soils, and it did reflect soil distributions in Alberta. Greatest in areas with brown Chernozems, which is this sort of washed out brown color in the middle of the province. Also, we found a lot in areas with regosol soils and solidized solonets soils. It was lower where there were tick abundance and prevalence was lower in areas where there was dark brown Chernozems, and again, our other group that included black Chernozems, dark gray Chernozems, district bruinsols, gray luvisols, humic vertisols, and some other groups. Because these features are often correlated, we used a presence or absence model fitted by stepwise regression to tease out which variables were most important in determining, or most important with the Dermacentor andersoni distribution. Our final model included weather effect, the temperature the previous summer, a couple of temperature the previous summer. Usually that was July, August, and September. A precipitation during the winter, January, February, and March, and also the difference between precipitation in the fall. And one of the problems we had was that autumn precipitation and winter precipitation were very highly correlated for these different sites. So by using this term precipitation differential, we were able to kind of get rid of some of the correlation and come up with something that fit a little bit better. Okay. When we looked at, say, plotting temperature in about increments, about 0.25 degree increments, we can see that, again, the ticks generally occurred in places where summer temperature was from about 14 degrees on up, and it tended to increase with that. Okay. Precipitation is plotted in groups of 5 millimeters, and tick prevalence increased where winter precipitation increased in the range 35 to 85 millimeters. We attributed this to the insulating effect of snow. The precipitation differential was used to incorporate autumn precipitation without introducing collinearity, as I mentioned. And basically, the wetter an area was in the fall, the more likely we were to find ticks there. We validated the model using read-one-out cross validation. All these statistics here, the agreement, sensitivity, positive predictive value, specificity, and negative predictive value, would indicate that model agreement was generally pretty good. When we plotted our model, we saw that the ticks really had largely a C-shaped distribution to the province. This is for 2011. This is for 2012, where abundance was greater, so it seemed to expand that much more over similar areas. Here's the little depression, sort of in the pre-foothills area, which has a fairly erratic snow cover during the winter time. And then what was interesting also was that we did see these little hot spots that went along, again, all up in the northern extension. That was the area where the tick had been described as occurring previously, but we were unable to sample during the set of studies. So I think it gave us a pretty good grasp of what was going on with andersoni in the province. So after that, we turned our attention to the Western Canadian distribution of both of these, and our results that I'll summarize now appeared in this paper, which just came out in 2025. We reduced the number of sites we did per year in Alberta and increased the number of sites that we had in British Columbia, Saskatchewan, and Manitoba. And we sampled from about 86 to 275 times per province over a 6-year period. So in total, we sampled 715 times at 454 unique sites. We found Dermacentor andersoni in Saskatchewan, Alberta, and British Columbia, with prevalence greatest in Alberta and British Columbia. Dermacentor variabilis was greatest in Manitoba and Saskatchewan, and we found a couple of instances where they occurred in both Alberta and British Columbia, and we'll get into that a little bit more later. What was also kind of interesting was that recently, in about 2016 or so, Dermacentor variabilis had been split into 2 species, and the western form of variabilis has been Dermacentor similis. And we did find, we went back to our samples that we had originally identified as variabilis from British Columbia and found that they were, in fact, a number of them were these Dermacentor similis, and we did pick a few of them up in there as well in British Columbia. So our new distribution for Dermacentor andersoni looks like this. It includes the gray areas, which are areas, again, where we found both Dermacentor andersoni and Dermacentor variabilis. And you can see that these 2 species form a nice little triangular area of distribution as their area of sympatry has expanded a little bit. We can see that Dermacentor andersoni is now just a little bit past that 105 degree western limit, occurs out to about 103. You see that it goes all the way up in through Alberta and again up into British Columbia. We did find one little spot here in the Creston Valley where we did find variabilis and andersoni together. So how that looks like if we look at it from geographic trends is that the prevalence of Dermacentor andersoni showed gradients with latitude, longitude, and also with elevation. These points or averages are observations grouped by ranks of the independent variables so that we would try to get 20 or so consecutive values into each plotted point. We can see that prevalence was greatest at latitudes south of about 51 degrees, and then it declined as we went northwards. The most northerly record that we had in that eastern Alberta was 52 degrees, 52.38 degrees. And again, that's slightly more north, about a degree more north than had been seen previously. With longitude, we again see a little bit of an expansion past this 105 degree magic number that we had before, but typically peaking again throughout Alberta and British Columbia. And then we saw that the prevalence really increased with elevation up until about, we get to about 1,000 meters and then declined somewhat, probably because of cooling weather. It's kind of important to note that the tick will occur at higher elevations as you move farther south to its distribution, and its distribution distributed right down into Mexico. So for Dermacentor variabilis, its western distribution now looks like this, very, very frequent in Manitoba, all over the place in Manitoba. Widely distributed throughout Saskatchewan, again, well past 105, comes right up and now extends into Alberta. So in the past 10, 15 years, it's basically moved from 108 degrees latitude past 110, about 110 and a half degrees or so. We did find one record of it again in the Creston Valley. And here we're looking at these little orange stars are basically the two areas where we found, oh, I see you're popping up there. We found the ticks that distributed in this Dermacentor similis in British Columbia. So again, here's what it looks like with the latitude, again, extending well past the 52 degree limit that had been done before, up to about past 54 degrees, 54.78 in Manitoba. We found it at 53.37 in Alberta. For longitude, again, our longest record, our biggest record was 116 in Alberta, and again, 110.88 in Alberta. So it seems to really be continuing its western expansion. Interestingly, though, for elevation, we found that the maximum elevation that we got at was at about nearly 1,200 meters, but it certainly does extend well past the 600 meter or the 500 meter limit that had been proposed earlier. Again, we use binomial regression models to sort of tease out the relationship of its distribution to other environmental variables. We see that, again, winter temperature was very important for both species, and other factors like summer and winter precipitation were also important. Summer precipitation played a big role with Dermacentor andersoni, less so with variabilis. We did find some effects of soil regions and soil groups as well. But just to go over this, I'll try to do this fairly quickly when we look at winter temperature, and I'll just focus on winter temperature, for example. We see that Dermacentor andersoni was distributed between -10 and a little bit between 0 degrees, and was most abundant right around, most prevalent around -5 degrees. With Dermacentor variabilis, I was really kind of surprised by this, that its upper limit that we found was at -5 degrees. And most of the ones that we found most of the instances where we found the greatest prevalence were in areas where it had really, really quite cold, which, -10 to -12 to -13 degrees. And that to me was really surprising, because this is a tick that does occur all the way through the southern United States down into Florida. So it's interesting that it's developed the ability to tolerate these very cold temperatures. We found a significant effect of ecoregion, which occurred basically for Dermacentor andersoni. The ecoregions on its most western distribution had a very positive effect on it. For variabilis, the ecoregions on its eastern side had a very negative effect on it. And these could just be due to secondary characteristics such as presence of hosts, soils, microclimate, vegetation, and so on. We also found a number of soils that really tended to boost the prevalence of them. Again, the Chernozemic, Solonetzics, and Regosols tended to elevate the presence of andersoni. One soil, Brunisol, the district Brunisols, tended to have a negative effect on distribution of variabilis. But again, the black Chernozems, Glasols, and Regosols had a positive effect on them, on variabilis. I just mentioned that we did, again, try to do some validation. Generally, we got pretty good values for agreement, sensitivity, specificity. Low values for positive predictive value. I would like to have seen these a little bit higher. And what positive predictive values really mean is that if you say that if your model predicts that you have a positive value, what's the probability that you're actually being correct there? And so here it's about 54%. On the other hand, we were pretty good at predicting negative values. So if we said it was negative, there was a 95% chance that it was a true negative. We did make some preliminary maps. This was just with the 2014 to 2016 data, so they're by no means cast in stone, and they're subject to a bit of, they'll be subject to revision as we add new data. With andersoni on the top, our map, again, pretty much predicts what we're seeing, a little bit more with the regular distribution of it. A lot of it mostly in these dry grassland areas through the interior of British Columbia and a little bit along the front of the Rockies. We need to revisit this a little bit more to see what we can get with this front range of the Rockies up in the more northern areas. One that's particularly interesting, though, to me was Dermacentor variabilis, where we can now see that it has very favorable habitat throughout much of Saskatchewan and Manitoba. And in terms of Alberta, we've got a fair bit of favorable habitat right in this area, in the east central portion of the province, where we have been picking it up. So it looks like these are areas that we should probably pay some particular attention to in terms of its future establishment. So just to go over a little bit of a summary, we have Dermacentor andersoni has had a largely stable distribution with a slight east and northern expansion. Dermacentor variabilis is continuing its western and northern expansion. We don't really know where it will stop, as it does seem to have a large amount of potential habitat. It's unclear why this species seems to be expanding more than Dermacentor andersoni, although we do consider it to be a very mobile tick species because it's frequently found attached to and moving on companion animals, particularly dogs. It's very strongly associated with human-assisted dispersal. The distributions of both species are strongly associated with climate, particularly temperatures during January and March. The ranges in these variables overlap between the species, but variabilis seems to be able to exist in quite colder areas, again, suggesting further opportunity to expand. We treated ecoregions and soils as modifiers in these studies. Their role is uncertain, but may have to do with direct effects on either ticks themselves through pH, moisture, or shading, or through indirect effects such as their influence on host abundance. There's still a lot of opportunities, I think, for further work with these. What we've done is generate some hypotheses, which is a benefit for this exploratory type of work. Some of these relationships could be tested using experimental approaches, and a good example of this was done recently, where Dyes et al. Looked at the actual effects of snow cover on off-host survival of variabilis. We've mainly focused on modeling prevalence. We really haven't done much with abundance yet, and we hope to continue on with this. We're starting a collaboration with the University of Calgary to do this. And finally, we're also planning on looking at examining how climate change may affect the distributions, again, with our collaborators from the University of Calgary. And that's it.
Hey, thank you, Tim. Great presentation. While I'm just doing a quick wrap up here, I just want to let everybody know that our last seminar of the series is going to be on Tuesday, the 21st. We're going to be hosting Dr. Diana Strahlenberg from Natural Resources Canada, who will be presenting on modeling and mapping refugia from wildlife and drought across Western Canada and what the implications of that are for climate-informed forest management and landscape planning. If you're not on the list, our mailing list, and you'd like to be added, please follow the link that Kristen's just added to the chat. And with that, I'd really like to thank Tim for his great presentation today, taking us through everything from the basics and the distribution of these two, I guess, different species of ticks that I'm used to thinking about. So a lot of things to think about that you're leaving us with, Tim. So thank you so much for a great presentation. Lots of folks in the audience giving you claps and kudos. And thanks also to everybody who tuned in. We'll see you next time.